this from campus. Okay. So you're muted, River.
Oh, I just say I'm recording this lecture and I will upload it.
Yeah, it's okay. Okay, so after extracting the DNA, you end up having like a soup with all these different types of DNA. So it can be from fungi, bacteria, it can be human, animal, anything is basically in there. So what can you do next? What is the next process that you would do? There are a couple different options. One is a normal PCR. So with the PCR, if you know exactly what you're looking for, like what kind of gene or what kind of organism you're looking for, you can target that. And basically with the normal PCR, you can just amplify the piece of DNA you're looking for and detect it. So detect it and amplify it. But it really needs that you know what you're looking for. So so you should have the primers for that DNA, for that piece of DNA you're looking for. Another thing that you can do is a qPCR, which is what we're going to do next week, which is basically the quantitative PCR. I'm gonna, you already have these in the videos. I'm just gonna briefly talk about the differences between the qPCR and normal PCR in the next few slides. But with a qPCR, you're basically quantifying that a specific piece of gene. So you know what you're looking for, you just wanna know how many you have in there. Another thing that you can do with the extracted DNA is doing a sequencing. So with the sequencing, you basically get the different, like the sequence of the DNA. So for each pieces of the DNA that you'll retrieve after the DNA extraction, you'll get the nucleotide bases like A, C, G, T, and the DNA sequence. And then afterwards, you can go to a library and basically try and find out which sequence of DNA is corresponding to which microorganism. So these are some of the stuff that you can do. So with the sequencing, you don't need to know what you're looking for. You'll get an idea of everybody that is in there. So it again goes back to what kind of question you have when you want to choose between these different options. So just a brief introduction about the qPCR that you're going to do next week. So basically, qPCR is very similar to qPCR and the PCR, very similar to a process of DNA synthesis. So what do you think you'll need for the DNA synthesis? Anyone? The answer is basically here in the figure. No one?
So wait, would you need oh sorry.
No, you go ahead. I'm sorry.
So are you asking like everything that you need to add or just the DNA polymerase?
Yeah, exactly. Like what do you think you need for the DNA replication? Yeah, as you said, one is the DNA polymerase. Then
Yeah, so you would need, I'm trying to think back to BioE 120. You would need the polymerase, extra nucleotides, and the DNA that you want to test for.
exactly. Yes, perfect. Yeah. You're gonna need the DNA polymerase, which is basically the enzyme that synthesizes the DNA. And then you also need like the nucleotides, these base pairs that you're gonna add, so that the DNA polymerase is gonna use to synthesize the DNA. You're gonna need the parent DNA, the target DNA that you're looking for, and the main difference between a PCR and a qPCR here is the detector. You're going to use a detector when you're doing a qPCR. One of the most commonly used detectors is cyber green, which is basically a fluorescent dye that binds to the double-stranded DNA and emits light. And the thermal cycler machine can detect that, and then we can help that with the help of the cyber green, we can quantify the number of pieces of genes that are being produced. So I'm gonna go, yes. So again, you have this in your slides in the video. I'm just gonna briefly mention that. So during the qPCR, you have, for example, this is your DNA template. This is the piece, the target piece that you are looking for and you want to quantify and amplify. So you'll do, so after setting up everything in the tube, you'll load everything on the qPCR plates you're gonna see next week, and then we put it in the thermal cycler machine. So what the thermal cycler does is that it will change the temperature in increments, like the time intervals that we specify for it. And then it repeats these cycles. So at the beginning, it will increase the temperature to 95 degrees. So this is like a rough idea, but it's around 94 to 98. It can be any of these. So when it increases the temperature, the double helix of the DNA, it unwinds and denatures. So the double strands become like two single strands. Then you're gonna have, then the thermal cycler decreases the temperature. They call it annealing temperature, where the primer at this temperature can bind to the specific part of the DNA that it is designed for. So the primer was this short single strand of DNA, which is complementary to the piece of DNA, to one part of the DNA that we want to amplify. So it basically goes and attaches to that part of the DNA. Then the DNA polymerase, which was the enzyme synthesizing the DNA, will bind to the primer. And using the nucleotides that we have provided, the DNA polymerase starts synthesizing that part of the DNA. So this cycle repeats. So at each cycle, like you're gonna have from one strand of DNA, one piece of DNA, you're gonna have two, then you're gonna have four, then eight, and that just increases rapidly. So at the end of each cycle, the qPCR machine will basically detect the cyber green emission. So the cyber green will bind to the double strands of DNA and emits light, and that is what the machine detects at the end of each cycle. That's very slow. So this is what happens basically. So your the thermal cycler machine is detecting the fluorescence at each cycle. So at the very beginning, you have only like one or two pieces of DNA. So the fluorescent dye, it's not that much, the fluorescent emission. And so it will at each cycle, as the DNA replication happens, the number of the amount of cyber green emission increases as well. So at one point, at one cycle, for the first time, the machine detects the emission light. So basically, that's the first, the CQ value. They call it the CQ value when the detector first sees the emission light, like the fluorescent dye. So if you, I'm waiting for this to go to the next slide. This is super slow. I'm sorry. This is my very old laptop. Okay, so For the qPCR, another difference is that we can provide known concentrations of that piece of gene that we have to produce a calibration curve. So at each run, you're gonna see next week in the lab that we actually provide the different concentrations of the piece of gene that we are looking for. So it's going to act as our standards. And then we're going to have a calibration curve. And the machine will give you an R-squared value, which is basically a regression of like shows how linear our calibration curve is and how precise our quantification is going to be based off of this calibration curve. And it's also going to give us an efficiency number, which is basically the idea is that ideally from one strand of DNA, one molecule of DNA, at the end of each cycle, you're going to have 2. So it should double every time. If that happens, you're going to have like the efficiency equal to 100%. But this doesn't happen all the time. And it can be due to different reasons, like if you have inhibitors in the sample, if you have, if the primers are not good, they're not binding perfectly, if you have a lot of primers in the sample, and for other reasons, you might have the efficiencies below or above 100%. But the perfect range is like 90% to like 110%. Oh, it doesn't go to the next slide. So basically the next slide is, if it works. The next slide is the questions for the assignment. I'm going to talk about those basically.
Yeah, or I guess you may create the presentation mode. You just select the slide from the.
That's what I'm trying to do. It is very slow. Like, yeah, this is my like my actual laptop didn't work for some reason. The Zoom didn't work, so I had to go to my yeah, it's very slow. I'm so sorry for that. So this is the slide I'm talking about. So the assignments, it would have been probably better if you had already started working on the assignments, but you probably haven't. I'm just going to briefly talk about it, but then you can come back to this presentation if you want to later on and like check it. So for the questions, the group assignment, you have 2 questions about qPCR. So one is related, the question A is basically related to the stuff you're going to do in the lab. It is the results that you get from the qPCR that we run for you in the lab. But question B is, it's not hypothetical, it's what I've done in the lab, in my own experiments before. So I just provide you with the numbers. I'll suggest you do the question B first. It is the simplified version of question A. So the idea is that we'll give you, so there are soil samples that we have collected from 3 different locations in the site, and we are looking for a specific bacteria, this time the Elephocoides. And these are the qPCR results, basically. So you have the gene copy numbers per qPCR well. You have the gene copy numbers, and you also have like how much DNA was extracted. So you had like 250 milligram of soil, extracted 100 microliter of DNA, used 2 microliter to do qPCR. You basically just have to back calculate and find out how many gene copy numbers of that piece of gene we had in our original soil sample, like per gram of soil sample. Here you're going to assume that you have your soil sample is 100% dry because we don't provide you with a moisture content. For question A, the difference is that you have the moisture content from your own experiments. So you're gonna have to account for that. And then the other difference is that we're gonna provide the numbers to you exactly as we'll get it from the qPCR machine, which is in nanogram. And then you just have to measure, calculate how many moles and then how many gene numbers you have in one well, and then calculate in per gram of soil. So it's basically that. So base pair is the number of nucleotides, and an average weight of a single base pair is this much per mole, and then you can just use that and know that the piece of gene that we have amplified is 165 base pairs. So just use these numbers. So I'm actually gonna go to, I'm gonna stop sharing for a second and go to your questions. Do you have any questions so far, by the way? All right, so I'm just looking at the ELMS, the discussion page, and I'm trying to see which questions we had. So the first question I see is from Amir. He has asked, how can we deal with filamentous growth problems in the treatment plant? Okay, from the video, does anybody remember if are filamentous bacteria all the time, like, are they all bad? No. So yeah, we need some of this, actually some of the filamentous bacteria. Do you remember what are the benefits of having filamentous bacteria in the active sludge?
They help with forming flocks, right?
Exactly, yes. So they help with forming the flux. The flux will basically help with separating the solid material and the water, the cleaner water, and also they will provide the solid structure for other microorganisms to grow. So you're gonna need the filamentous bacteria, but there are so many different types of them. There are some types that are actually not beneficial as much. They can produce foams that would affect the efficiency of the sludge treatment, or also the excessive production of the filamentous bacteria is not good either. So to my understanding, one of the ways that they deal with that is basically using some polymers or coagulants, addition of polymers or coagulants to decrease the amount of filamentous bacteria. They also do chlorination, so they add colorings. The chlorine can affect everything, but for the flux, it mostly affects the surface of the flux, so it doesn't penetrate as much into the flux, so it basically kills the bacteria outside, but the rest can survive. Also, the long-term solution to it is to basically just monitor the wastewater and the nutrients that they add and the changes in the aeration and everything. So they have to, for each plant, they have to find their sweet spot on their own. So they have to monitor everything and make sure that everything is the right place.
Thank you.
The other question, what kind of microbiology research is being completed related to drinking water, wastewater at University of Maryland? So that's an interesting question. I work on bioremediation of groundwater, basically. So I'm looking for different microorganisms in the groundwater that are capable of degrading contaminants, certain types of contaminants in the groundwater. Also, there are other people in our group that were working on basically these microorganisms that are able to deal with contaminants in the wastewater treatment plants. So the ones that are able to degrade the toxic chemicals, basically. There are also like people work with the same concept in Davis's group. I think that Dr. Davis's group for stormwater. And I think Dr. Lee groups also, they do a lot of wastewater stuff. Lou's not here. She would have been able to explain a little bit more about that. But yeah, these are the type of stuff that we do. Hope I have answered your question.
Yeah, it did. Thank you.
No problem. So during the growth of bacteria in generations, if an outside factor caused a small cells to die, what would the growth rate constant look like? What do you guys think? What happens if an outside factor caused bacteria to die? How does it affect the growth rate?
Probably like, oh, sorry, go ahead.
No, go ahead.
I was just gonna say it would maybe be like a discontinuity in the graph.
Yeah, it very much, I think, depends on how that factor is actually affecting the bacteria. So if it like kills off all the bacteria, of course, or if it it affects it in a way that the number of cells that are being produced are less than the number of cells that are dying, then you'll get to the decay process or the death phase. And then you can use like the decay coefficient to like calculate for that. So you're gonna have the death phase basically. But yes, if it is basically if still the number of cells that are being produced are still higher than the number of cells dying and you're still in the exponential phase, we'll probably just decrease the growth rate, right? Another question, have past diseases been tracked through wastewater? Very interesting one. So yeah, I did a little research. Mostly they do the diseases related to like intestinal problems. Usually they do a surveillance for that. So like parasites or seasonal dynamics and some kind of types of viruses. They use wastewater surveillance for that, or even salmonella outbreaks. Also, I even encountered like I even found that people use wastewater testing for illicit drug use in prisons. So they want to like track how prisoners actually do. And another very hot thing that they do with like wastewater surveillance is antimicrobial resistance genes that they try to track down because we consume so many compounds that have antimicrobes in them. So we basically, in the wastewater treatment plants, there's a very good condition that these can be actually studied and see how it has affected the whole community, basically.
Thank you.
No problem. Are there microorganisms seen in wastewater that is an indicator that something is absolutely wrong, other than the example with nematodes and rotifers? So basically, like we talked about, so if you have an excessive amount of filamentous bacteria, that's not good. Anything off balance is not good. So you usually like to have diversity. So diversity is basically the key. These are acting as an ecosystem, right? So everybody has a role. So you have to find the balance. And if you have something in an excessive form, whether it is a bacteria, whether it is fungi or other organisms, is usually not good. And it shows that we have to take action. The other question, how does the composition of microorganisms change with the environment of activated sludge system? What happens when the dissolved oxygen levels are too high or too low? Okay. So, for example, one thing that they use for wastewater treatment plants usually to track everything is the ratio of food to microorganisms, food to mass ratio. For example, if, yeah, different conditions definitely would affect this activated sludge, but if you have, for example, a high food to mass ratio, biomass ratio, you're gonna have high concentration of bacteria because those are the ones that are capable of reproducing very fast. And so you're going to have high concentration of bacteria, so the more complex compounds are not being degraded because the other microorganisms are not there yet. So you're going to get low flux or poor settling happening. Or if you have low food to biomass ratio, you're going to see that the bacteria clump together, but they're still dispersed. So, and you'll see some types of filamentous bacteria increasing in the wastewater. If you have low dissolved oxygen, for example, some of the filamentous bacteria will increase. It will cause the imbalance, of course. And if it gets too low, basically you're going to have a very low concentration of bacteria, therefore you're going to have a turbid flow, very low quality, very low effluent quality. If you have high dissolved oxygen, it's usually good, but if it is excessive amount of oxygen, then you're going to have shearing the flux, dispersion of the flux, resuspension of the flux, and less denitrification will happen at that condition. And also, for example, another condition change, another parameter is like the pH. If you have very low pH, you're going to have excessive growth of fungi. So these are all the parameters, yeah, that they have to like take into account when they are monitoring the process. What purpose do tardigrades serve in the activated sludge? Anybody? Any guesses? What are they? Are they animals? Are they single cellular? But what are they? Tardigrades are actually animals, so they're a higher form of life, and they basically eat everything. So they can eat cells, they can eat algae, they even prey on other tardigrades, so they do cannibalism. They eat rotifers. Maybe that's why they're so resistant, because they can basically eat anything. So their role is to keep the balance, right? So it's still like an ecosystem. You want to it's like any other ecosystem. If you don't have the predator, then like you're gonna have an unbalance in the ecosystem. How are chemostats able to keep growth in the exponential phase if you don't know the growth rate in advance? Well, the idea with the chemostats is basically very simple, and that is you're keeping everything in a steady state. And in the steady state, in this condition, the specific growth rate is actually equal to the rate at which the culture is being diluted. So you're adding like the new flux of nutrients and everything, and the waste products are getting out. And at this, so the specific growth rate is basically equal to the rate of the dilution. So by varying the dilution rates, you can basically establish the steady state and at different growth rates, basically. Next question from Emily. Lab yesterday, many steps, we ended up with brown color palette. Did the steps we took during extraction impact color? Does dyeing impact the cell's color, or just that they are now all clumps together? So no, actually no, the cells dyeing doesn't impact the color. What you see in terms of color is mostly from the soil, and it is mostly like the humic acids and all the other organic materials that you see in the soil that would affect the color of the pellets. When taking water sample, I didn't get this part of your question, Emily. Can you a little bit explain? Do you have to perform some sort of rinse of the bottle to ensure all the bacteria has been
Yeah, I guess when I was like when I was watching videos and stuff, I was just thinking about like when the bacteria start to grow on the sides of the bottles. I work in a lab where we process water samples, and we never rinse the sides. And I was just thinking, like, since bacteria kind of likes to form like biofilms, you know, like in your lab, if you ever process water samples, do you guys like somehow rinse the bottles and to make sure you're processing all of it?
Again, I'm not sure I quite understand. Are you talking about like what is the purpose of what kind of process are you talking about?
Honestly, it's okay. You can just go to the next question.
Yeah, it's okay. Okay. So yeah, bacteria tend to form biofilm, depending on what you're doing. If you mean the turbidity measurements, for example, for the measurements that are with for the turbidity measurements, you usually try not to use the bacteria that tend to form biofilms as much, because that would affect the turbidity measurement. But in general, yeah, I would say it really depends on the experiment, whether or not you want to mess around with the bacteria. Sometimes you even benefit from the biofilms being formed. Yeah, it really depends on what you want to do, actually. The next question, at the wastewater treatment plant, they seem to look for indicator organisms rather than the actual bacteria. Why is that? So in wastewater treatment plant, it is so if they want to do like a qPCR or things like that, it is not like efficient at all, because it takes a lot of time. It needs like specific trainings, and it also like needs like expensive equipment and a lot of time to do that. So they would use other indicators, usually just microscopy and other larger microorganisms, because they're doing microscopy instead of looking for the bacteria. They usually use the larger microorganisms that can be detected. Does that answer your question? And the mu and QS of the monod model, how is it? So yes, exactly. It has to be calculated for each system, meaning that this is empirical. So you have to, for each system, they have to go back and set up different substrate concentrations, measure the growth rate, and then have that and like draw that graph and find the mu max and find the KS values, basically. So that's kind of empirical. And from there, then they can like find the other parameters. The sludge age and HRT are not the same. Yes, they're not the same. HRT is the hydraulic retention time. That is the time that the wastewater passes through the tank. Whereas the sludge age is the time where the bacteria are under the aeration tank. But it is odd that they are both represented by theta. Honestly, I'm not sure if they're both represented by theta, but I've just seen mostly people writing HRT and SRT, but yeah, I know that in the slides we had theta for the sludge age. But yeah, it's just like the symbol. I don't think that matters that much. The important thing is that you know that the sludge age is different from the hydraulic retention time. We use the sludge age mostly to maintain the proper amount of activated sludge that should be inside the aeration tank at each specific time. Other questions? Did anybody have any other question? Why am I? Oh, I see another one. That for substrate utilization, is it possible that the growth rate is not a first order reaction? Yes. So the first order reaction would be when we are calculating everything in a batch reactor, but if you have a full through state, like if you have other conditions, that might not be a first order reaction anymore. I have some links to the growth kinetics, like how it is calculated. So there are other models for calculating the growth rate if it is not a batch reactor system that can be used basically, but yeah, they're more complicated usually. So I put a lot of stuff, a lot of useful links that I found on the slides that I'm gonna share with River. He's gonna upload it, and then you might be able to see some of those. Any other questions, guys?
If you send me, I can upload it on else.
Sure.
So any more questions? Okay, and I think I have 2 points to add to Edmy's question, like because Edmy also asked like if we have some biofilms attached on the surface of the tube, that is not good. As a outsider of microbiology, I haven't do those kind of experiments, but I would say that if many people use that method, I think that would be good. But I also think there might be some biofilms that will form attached to the surface of the tube, which may not be easily removed or transferred when we are doing the transferring experiments. But I would say this could be one of the errors that introduced in this experiment. So I think if we are interested about the errors or how can we improve the accuracy of this method, we may think about this. And for the next one is about the indicator organisms. I would say that indicator is very popular in environmental engineering. And except for microbiology, we also do some indicator chemicals in like chemistry. Like if we are interested in some toxins in our body, like it may have different forms. So we may just find one toxin that is relatively easy to measure. And then if this easy toxin has a good correlation or linear relation with other toxins, then we can measure this only one toxin, and then we can accurately predict the concentration of other toxin. By using these methods, we can save time and we can save money and we can get the fast result. So I think this is the same for the microorganisms. If we can find one organism that has a good correlation between other organisms, then we can just simply test this one organism with smaller time and with inexpensive methods. We can get a good prediction or accurate prediction of other bacteria that can also save some money. Yes.
Yeah, exactly.
Yeah, so any more questions? If you guys don't have question, I guess we can, that's all for it today. And again, so for the next Tuesday, we are going to the class. And that's it.
See you guys in the lab next week. Bye.
Bye.
Thank you.
Thank you.
Thank you.